Good day, juniors. Last week you discussed about hematopoiesis and the principles involved in hematopoiesis. Also, you discussed the different stages of hematopoiesis starting from the embryonic stage. Also, it was discussed to you the different changes that occur to the RBC during its developmental process, what happens to the nucleus and to the cytoplasm of the cell, the changes that occur to the nucleus and to the cytoplasm during the maturation process of the RBC. And also, it was discussed to you the fate of the RBCs when they are subjected to a normal destruction process. Now, for this lecture, we are going to discuss the different metabolic activities that RBCs perform in order to carry out its function more efficiently and for the survival of the cell in general. Your RBC is also called erythrocyte. It has a size of 6 to 8 micrometer with an average of 7.2 micrometer. What is the shape of the RBC? Yes, biconcave. Its lifespan ranges from 90 to 120 days, and the very function of the RBC is to transport gases. It delivers oxygen from the lungs to the tissues, and it carries carbon dioxide from the tissues to the lungs to be exhaled. Now, this oxygen carrying capacity of the RBC is because of a protein that is abundant in the cytoplasm of the RBC. This protein also gives the RBC a red in color. And this protein is what we call, yes, hemoglobin. When hemoglobin is bound with oxygen, it is called oxyhemoglobin. But when it is devoid of oxygen, it is called deoxyhemoglobin. Oxyhemoglobin is bright red in color, whereas the oxyhemoglobin is dull red in color. Your arteries carry oxygenated blood, and because it is oxygenated, it carries oxyhemoglobin. That is why your arterial blood is bright red in color. Your veins, however, they carry the oxygenated blood, and therefore they carry the oxyhemoglobin. And because of that, your venous blood is dark red or dull red in color. The shape of the RBC is biconcave because it has two concavities. One concavity on this side here and another concavity on the other side. Now, this biconcave shape of the RBC allows the RBC to efficiently load and unload oxygen. Now, when we are going to view RBC using a stained smear, your RBCs look like this. Okay, the RBC has a darkly stained periphery and an area that is minimally stained or unstained. This central area that is minimally stained is what we call the central pallor. Central pallor because it is pale, and this central pallor is about one-third of the diameter of the cell. Now, it is important that the central pallor is maintained at a one-third only because a bigger central pallor indicates that there is something wrong with your RBC. Now, the darkly stained periphery of the RBC is because of its hemoglobin. Most of the hemoglobin of the RBC is concentrated at the periphery. Now, what is the importance of this? Because of this characteristic that the hemoglobin is concentrated at the periphery, it is easier for the hemoglobin to uptake and to unload oxygen at a faster rate. How so? Now, when oxygen diffuses into the cell, it will readily bind to the hemoglobin because the hemoglobin is already there at the periphery. So it takes a lesser time for the oxygen to bind with the hemoglobin because the hemoglobin is already found at the periphery. When the hemoglobin is concentrated at the center, it will take more time for the oxygen to bind with the hemoglobin because the oxygen has to travel from the periphery then going to the center. So it takes a longer time for that. Now, but because the hemoglobin is concentrated at the periphery, therefore it is easier and it takes lesser time for the oxygen to bind with the hemoglobin. Likewise, during unloading, it will also take a lesser time for the RBC to unload oxygen because the oxygen is already present at the periphery. So it takes lesser time to load and to unload oxygen when the hemoglobin is concentrated at the periphery. And this is an important characteristic of the RBC because remember, the function of the RBC is to efficiently transport oxygen to the tissues where it is needed. Therefore, this transport processes or this loading and unloading of oxygen should not take much time. So it should be rather at a faster rate because your tissues are constantly in need of oxygen. So your RBCs must be able to supply oxygen at a faster rate. Okay. Now, your RBCs have a volume of 90 femtoliters and a surface area of 140 micrometers square. It has a very thin cell membrane. This thin cell membrane allows diffusion of substances, including gases, to easily diffuse in and out of the cell. Now, as you can see, the RBC has a large surface area compared to its volume. Now, what is the importance of having a large surface area compared to the volume? Okay. Now, most human cells, when human cells increase in size, their volume also increases. Uh, it's like they become cube. No? It's like a cube. When the cube increases, the volume inside also increases. Now, when it comes to RBC, it doesn't go that way because RBCs have larger surface area compared to its volume inside. Now, what is the advantage of this for the RBC? When it comes to diffusion, it is an advantage for the RBC if it has a large surface area than its volume. How so? Now, I have a good analogy for that. Imagine a room with the same size, for example. But one room has only two doors, while the other room has about 50 doors. Now, both rooms must be filled with 100 people. Now, between two rooms, which room would be able to fill it, to be filled in more faster than the other? Of course, exactly. It's the room with 50 doors. It will be easier to fill in the room with 50 doors because there will be more doors where people can pass through. Whereas the other door with two doors only, people have to line up, people have to get somewhat, they will be stranded because of the very slow traffic in their entry to the room, right? So the principle is uh, sort of like that when it comes to the RBC or to the cells having large surface area. When the cells have large surface area, the rate of diffusion will be a lot faster. And this is important for RBC because RBCs have to transport oxygen, have to deliver oxygen to the different tissues at an efficient rate. So in order for the delivery of oxygen to take lesser time, no, lesser time, the RBC has to have a large surface area compared to its volume. Okay. Now I have a GIF here. Look at what happens to the RBC when it unloads gases. So as you can see, it increases its surface area when it unloads gas. Okay. And then, so So whenever there will be diffusion in and out of gases, the RBC will increase its surface area for the diffusion to occur at a faster rate. The RBC membrane. The RBC membrane is composed of 52% proteins, roughly 52% proteins, about 40% lipids, mostly composed of phospholipids and cholesterol, and about 8% carbohydrates. Now, the lipid content of the RBC membrane is, as I have said, primarily cholesterol and phospholipid. Well, as we know, the phospholipid is responsible for the selective permeability of the cell. Now, the cholesterol, on the other hand, it maintains the fluidity of the cell. Fluidity, well, yeah, it renders the cell membrane um, flexible and also deformable. When the cell has a balanced amount of cholesterol, its RBC membrane is fluid, therefore it is not rigid. Likewise, the lipids in the RBC membrane provides tensile strength to the RBC. Now, what is this tensile strength? Tensile strength refers to the capacity of the RBC membrane to be pulled so much and be exposed to a certain level of shear stress without breaking. So in other words, the RBCs can stretch itself, okay, without breaking, then it can just go back to its normal shape as if nothing happens. Now, this particular characteristic is important when RBCs have to travel to the different tissues of our body in the very small capillaries. If it's very small capillaries, RBCs have to squeeze themselves into those small capillaries, and in order for the RBCs to fit in through those small capillaries, it has to stretch itself. Okay? But its stretching must be enough not to cause hemolysis of the RBC. And then after passing through those microvasculatures, RBCs will go out and then will go back to its natural shape as if nothing happened. Okay? Now, the proteins of the RBC, well, most of these proteins are skeletal proteins, and these cytoskeletal proteins render or maintain the shape of the RBC and also contributes to the flexibility and deformability of the cell. Now, some of these proteins are also transporters, no? These are channels for ions in order for ions to pass through the membrane of the cell. Now, these ion transport channels are important in order to maintain an adequate level of intracellular electrolyte. Now, the RBC membrane, as I have said, it is used to separate the intracellular environment from the extracellular environment. It allows nutrient and ion passage selectively in and out of the cell, and it also allows the cell to deform when required. And as I have mentioned, it is an important property of the RBC to be flexible and at the same time deformable, especially that RBCs deliver oxygen to the different tissues of our body. They pass through microvasculatures, very small capillaries. And in order for them to pass through those very small blood vessels, they have to change their shape, they have to stretch themselves. And then, as they pass through, they will just go back to their normal shape as if nothing happened. Okay? Now, this is a 3D representation of the membrane of the RBC. As you can see, there is an interaction of the different proteins. Okay, and then embedded also on the RBC membrane are the different proteins. So you have proteins that are integral or transmembrane proteins, which are embedded or which traverses the plasma membrane, and those proteins that are found inside the plasma membrane. So now let us discuss the interaction that occurs between these different proteins in the RBC membrane. The primary cytoskeletal protein in the RBC membrane is the spectrin. They come as alpha and beta spectrin, and they have a helical configuration. Alpha and beta spectrins are bound to another skeletal protein called ankyrin. Ankyrin is bound to band 3 protein. Band 3 protein is an anion exchanger. Now, the interaction of ankyrin and band 3 protein is stabilized by another protein called the band 4.2. Now, aside from this interaction, alpha and beta spectrin also interacts with glycophorin C. Now, this interaction is mediated by band 4.1. The presence of band 4.1 here also stabilizes the interaction of spectrin and actin. Now, I want to emphasize glycophorin C. What is glycophorin C and what is the importance of glycophorin C? Glycophorin C is a transmembrane protein. It acts as a channel for sialic acid to pass through. Now, the Glycopharin C is responsible for the negative charge surface of the RBC. Now, what is the importance of having a negatively charged surface? Your RBCs basically do not aggregate. They do not stick with one another because they basically repel one another. They repel one another because they all have the same charge. And remember the principle that like charges will repel. So one RBC will not stick with another RBC because they are both negatively charged and they will repel each other. Likewise, your RBC will not stick to the blood vessel wall. Hindi yan siya didikit doon because your blood vessel wall is also negatively charged. So, your RBCs will just flow through your circulation, through your blood vessels undisturbed without sticking with another RBC or without sticking to the blood vessel wall under normal circumstances because the RBC and the blood vessel walls are both negatively charged surfaces, so they will just repel each other. Now, these are other proteins, examples of transporter membrane proteins and their functions. These transporter membrane proteins are found on the surface of the RBC, either as peripheral or a transmembrane or an integral protein. Okay? Now, class, you have to remember that the interaction of the different cytoskeletal proteins in the RBC membrane maintains the shape, the flexibility, and the deformability of the cell. Any abnormality, okay, abnormality in at least one of those skeletal proteins will result to a disruption in the balance. And if there will be disruption in the balance, it will cause the RBC to become rigid. Therefore, they will become less flexible and less deformable. Now, when they are less flexible and when they are less deformable, they are easily subjected to hemolysis. Okay, and when they are, and that means that there will be premature destruction of the cell. Why premature? Because these cells have not yet served the entirety of their lifespan. No, they are destroyed very rapidly because they were not able to withstand the turbulence of the environment that they were passing through. For example, in the spleen has very small sinusoids there, but a normal RBC can just pass through effortlessly through those small sinusoids because the RBCs can can stretch themselves through those sinusoids and then will just go back to its normal shape after passing through. But for RBCs that have abnormality in their shape or RBCs that are less flexible or less deformable, they will have a hard time passing through those small areas. As they go through those small sinusoids, sometimes they become trapped there. And when there are RBCs trapped in the sinusoids of the spleen, the macrophages will be activated because the function of the one function of the splenic macrophages there is to remove anything that have been trapped in the splenic sinusoids. So the macrophage will destroy those RBCs that have been trapped there. And most probably, these RBCs have not yet served their entire lifespan when they have this kind of abnormality. So therefore, they are subjected to a premature rupture. Now, an example of an RBC having an abnormal shape is this, a spherocyte. Okay, kindly take a look at the difference in the shape of the normal red blood cell and a spherocyte. A normal RBC has a biconcave shape, whereas a spherocyte has a spherical shape. Okay, now this type of cell here cannot withstand osmotic pressure as much as a normal RBC. Now, what do I mean? Okay, example, balloon. Okay, you have a deflated balloon for a normal RBC and an already inflated balloon for a spherocyte. Now, again, our analogy is like this. The normal RBC is represented by a deflated balloon, while the spherocyte is represented by an already inflated balloon. Okay. Now, example, you are going to put water in those balloons. Now, which of the balloons will cater more or will be filled with more water? Is it the deflated balloon or is it the already inflated balloon? Of course, it is the deflated balloon that will be able to cater or to be able to hold more water. The already inflated balloon, okay, it's already inflated. Now, when you put water in it, they will burst rapidly. It cannot cater more water or it cannot cater water as much as the normal RBC because this kind of cell, okay, a spherocyte will not be able to expand as much as a normal RBC. The normal RBC, having a biconcave shape, can cater more water in it because it can expand a lot more. But a spherocyte, because it is already spherical in shape, it cannot expand as much as the normal RBC. And because it cannot expand that much, the cell will be subjected to a premature rupture. Again, I term this as premature rupture because this kind of cell have not yet served its entire lifespan before hemolysis. This kind of reaction, the hemolytic process occurring here was only possible. It's because of the changes in the osmotic pressure. No? Na this kind of cell may have not yet served up to 120 days of its lifespan. It was destroyed because it cannot cater much or it was not able to respond much or enough to the change in the osmotic pressure. Okay. Now, the components of the cytoplasm of the RBC is also important to maintain the overall property of the RBC. Now, these components or these contents of the cytoplasm involve the electrolytes, the metabolic materials such as glucose and the intermediate products of glycolysis, and the different enzymes that catalyze the metabolic reactions in the cell. Okay. Now, let's start with the ionic transport and the hydration homeostasis. Now, the hydration status of the RBC is dependent on the intracellular electrolyte content. Okay? Now, intracellularly, potassium is more abundant, about 140 MEq per liter, whereas sodium intracellularly is much lesser than the concentration of the potassium. Intracellularly, sodium is just about 10 MEq per liter. Sodium is more abundant extracellularly. Potassium is more abundant intracellularly. Now, why is it that there is lesser concentration of sodium inside the cell? Now, it is important to maintain low concentration of sodium inside the cell because the presence of sodium inside the cell will attract water. Now, if there will be more sodium inside the cell, more water will come into the cell, and if there will be more water that will go to the cell, it can cause the cell to swell, and then eventually the cell will burst. And when the cell is swollen, okay, when the cell is swollen, it will not be able to pass through the different microvasculatures because when swollen cells pass through small blood vessels, they will be ruptured there because they cannot squeeze themselves in there. So they will be destroyed in the process, no? So that is why intracellularly, sodium should not exceed. Okay, so sodium should not exceed intracellularly in order to avoid the accumulation of water inside the cell. Now, remember this also, class. The inward diffusion of sodium should not exceed the outward diffusion of calcium. Okay, I have to repeat that. The inward diffusion of sodium should not exceed the outward diffusion of potassium. Yes, potassium is abundant extracellularly, but potassium, rather, again. Potassium is abundant intracellularly, but potassium also has to go out of the cell at some point, okay? But the diffusion of potassium outside the cell should not exceed the diffusion of sodium inside the cell, okay? Now, when the outward diffusion of potassium exceeds the inward diffusion of sodium, it causes the cell to be dehydrated. Okay, again, let me repeat that. When the outward diffusion, palabas, when the outward diffusion of potassium exceeds the inward diffusion of sodium, the cell will become dehydrated. When there will be more and more and more potassium that will go out of the cell than sodium that will go inside the cell will become dehydrated because as potassium goes out of the cell, water follows potassium. So if there will be more and more and more potassium that will go out, more and more and more water will go out of the cell. Okay? So there has to be a balance in the inward diffusion of sodium, okay, to supply enough water. There has to be a balance between the inward diffusion of sodium to supply water and the outward diffusion of potassium to remove excess water. Okay? I hope that's clear. Now, there has to be a um, enough concentration of calcium inside the cell also. Intracellularly, calcium is much, much, much lower compared to the extracellular concentration of calcium. Calcium is not allowed to accumulate inside the cell because when calcium accumulates in the cell membrane becomes rigid. Okay? Now, in order to avoid accumulation of calcium inside the cell, the Gardus channel will be activated. This Gardus channel is a calcium-dependent potassium channel. Now, for example, there is accumulation of sodium inside, rather, there is accumulation of calcium inside the cell. And then the accumulation of calcium activates the Gardus channel. The Gardus channel will diffuse potassium out of the cell. Okay? It will diffuse potassium out of the cell, and in the process, calcium will be diffused out of the cell also. How so? This is because the Gardus channel is calcium dependent. So when an ion is exported out or in of the cell, the calcium will also follow in and out of the cell. So if it happens that the calcium becomes abundant inside the cell, Gardner's channel will diffuse potassium out of the cell, bringing calcium together with potassium. So, isasabay ni Gardner's channel pagpapalabas ang calcium together with potassium in order to prevent accumulation of calcium inside the cell. Okay? I hope that's clear. Now again, there has to be a balance between these electrolytes because these electrolytes will maintain the hydration of the cell, particularly inside the cell. Okay, so the ionic transport in the RBC cell membrane involves the passive leak through ion channels driven by the electrochemical gradients, a gradient-driven passive or secondary active transporters, and the active exchange through sodium-potassium pump and calcium pump. And as I have mentioned, intracellularly, potassium is more abundant than sodium. Sodium is not allowed to accumulate inside the cell because sodium will attract water inside the cell. Now, if water is starting to accumulate inside the cell will drive potassium out, papalabasin si potassium, because when potassium is diffused outward, water will follow with potassium. But there has to be a balance between the entry and exit of sodium and potassium. Okay? The RBC energy metabolism. RBC energy metabolism is a very important activity of the cell because, well, all of the cells require energy in order to carry out its function, right? Now, your erythrocytes, your RBCs, are basically enucleated, meaning they do not have nucleus. To review, the metamyelocyte or the orthochromatophilic normoblast is the last stage that is nucleated. After metamyelocyte, the next stage to that, which is the reticulocyte, it is no longer nucleated. Wala nang nucleus sa reticulocyte. Now, the, the transition between a metamyelocyte and reticulocyte also extrudes other organelles. Okay, the organelles together with the nucleus are extruded out of the cell. And this reticulocyte, as it travels to the spleen, okay, it undergoes polishing in the spleen, where the remaining organelles are taken out of the cell. Now, the nucleus and the organelles are extruded out of the cell because this allows the RBC to carry an abundant amount of hemoglobin because, as I have mentioned, the very function of the RBC is to transport oxygen. And for oxygen delivery to be efficient, there has to be an abundant amount of hemoglobin in the cytoplasm of the RBC. But losing the nucleus and losing the organelles of the RBC limits the metabolic activity of the cell. That is the reason why the RBCs do not rely on its organelles because basically it no longer has organelles. However, your RBCs rely on the different enzymes that catalyze the different metabolic reaction inside the cell. Now, for the energy metabolism of the cell, the primary energy metabolism of the RBC is through glycolysis. And of course, this kind of glycolysis is anaerobic, so it does not use oxygen. Okay? And likewise, there is no um, electron transport chain in our RBC because your RBC no longer contains the mitochondria. It has already lost its mitochondria, so no electron transport chain for the RBC. Only the anaerobic glycolysis, which is the Emden-Meyerhof. Now, in your biochem, you have already discussed the different changes that occur to the glucose molecule as the glucose is converted or as the glucose is metabolized in order to generate ATP. Okay. Now, glucose is the only carbohydrate that is readily metabolized by your RBC. Okay. Now, when glucose is metabolized in the process, okay, your glucose utilizes 2 ATP. As in here, it consumes 1 ATP and another ATP. So, your RBC consumes 2 ATP, and along the way, it generates 2 ATP plus 2 ATP. So all in all, your RBC for one glucose molecule, your RBC is able to generate 4 ATP molecules. But because of the consumption of ATP in the earlier part of the metabolism, there is only 2 net ATP that is generated in this pathway, the glycolytic, the Emden-Meyerhof pathway. So again, the Emden-Meyerhof, it produces 90% of glucose energy used by the RBCs. So it provides most of the ATP that is utilized by RBCs to perform its function. The Emden-Meyerhof, it consumes 2 molecules of ATP in the earlier stage of the glycolysis, and it generates 4 molecules of ATP all in all. But because it consumes 2 ATP, so there is 2 net ATP produced in this glycolytic pathway. Now, the energy generated during the Emden-Meyerhof pathway is of course used by the RBC in order to perform its function efficiently. So the RBC is able to deliver oxygen effortlessly, okay, under normal condition because of the enough supply of ATP. Likewise, the amount of ATP generated during the Embden-Meyerhof pathway is important to membrane the integrity of the RBC membrane. And I have mentioned earlier the importance of the integrity of the RBC membrane. When the RBC membrane is in good shape and in good condition, it will allow the RBC to become flexible and at the same time deformable. So it allows the RBC to go through very small blood vessels to squeeze themselves in through those very small blood vessels without causing destruction to the cell. Likewise, the energy generated will maintain intracellular electrolyte balance, and this electrolyte balance maintains the hydration status of the cell. And lastly, the energy generated will allow the RBC to live its entire lifespan, which is around 90 to 120 days. When the RBC is depleted of energy, of course, it will lose its function, it will lose its viability, and the RBC is subjected to premature rupture. Okay? So please remember that. Now we have the so-called diversion pathways. What are these diversion pathways? They are called as diversion pathways because, well, basically, they are diversion from the Emden-Meyerhof pathway. And the RBC has three diversion pathways that are of equal importance also. It is not the Emden-Meyerhof that is only important because all these other three pathways are also important in order to maintain the viability of the RBC. Now, these pathways are the hexose monophosphate pathway, the Lou-Behring-Rappaport pathway, and the methemoglobin reductase pathway. And we are going to discuss the importance of each pathway and the different processes involved in each pathway. Now, the hexose monophosphate pathway. It is also called as the pentose phosphate pathway. Okay, now what is the importance of this pathway? Now, every day, class, your cells, your RBCs, and your other cells are exposed to oxidative stress brought by oxygen-free radicals. What are these oxygen-free radicals? These oxygen-free radicals are basically byproducts of oxidation when oxygen interacts with molecules during a certain metabolic process. So during that interaction, there will of interaction of oxygen with other molecules, there will be a generation of oxygen-free radicals. And these oxygen-free radicals become dangerous when they interact with the cellular elements such as DNA. As in the case of our RBC, oxygen-free radicals become dangerous when these oxygen-free radicals interact with the hemoglobin, primarily the globin chain of our RBC. Now, um, so what is the mechanism of the RBC in order to prevent from being exposed to these oxygen-free radicals? Of course, by generating antioxidants. Antioxidants from the term itself, anti against oxidants. So these antioxidants are the ones that will interact with those free radicals. So these antioxidants will prevent the oxidation of our hemoglobin. And that is when the hexose monophosphate pathway comes in. Because your hexose monophosphate pathway protects hemoglobin from oxidative stress by generating NADPH. Okay, so now what happens in the process? Okay, your glucose 6 phosphate, remember class had that this is a diversion pathway from the glycolysis. Remember in the earlier part of the Emden-Meyerhof, glucose is converted into glucose 6 phosphate by the enzyme hexokinase. So that glucose 6 phosphate, some of those glucose 6 phosphate will proceed to the hexose monophosphate pathway. So what happens in the hexose monophosphate pathway? Your glucose 6 phosphate in the HMP, okay, is acted upon by the enzyme glucose 6 phosphate dehydrogenase. Again, G6PD, glucose 6 phosphate dehydrogenase. This enzyme requires the presence of NADP as its cofactor. So in this process, when glucose 6-phosphate is acted upon by G6PD, NADP becomes reduced NADPH. Okay? Now, saan galing yung H? The H there comes from the glucose 6-phosphate. Okay? Now, when the glucose 6-phosphate is removed with hydrogen, glucose 6-phosphate becomes 6-phosphogluconate. Again, this process is mediated by the enzyme G6PD. And in this process of converting glucose 6-phosphate into 6-phosphogluconate, NADP is converted to its reduced form, which is NADPH. Now, this NADPH will proceed to the next step. And the next step involves the glutathione. Now, what do we know about glutathione? Yes, indeed, glutathione is an antioxidant. So, in this next step here, the glutathione will be acted upon by glutathione reductase. And in the process, the glutathione reductase requires the presence of NADPH as its cofactor. So, because NADPH is consumed, it is converted back to NADP. And during the reduction process, glutathione is converted to its reduced form, which is the reduced glutathione. Now, what is the importance of this reduced glutathione? Reduced glutathiones are the ones that will interact with the oxygen radicals. Example, peroxides. Peroxides are harmful to our cells, not just to our RBCs. Peroxides can cause oxidation of our cells. Now, in order to prevent this oxidation from happening, the glutathione will act against these peroxides. So, peroxides, once acted upon by reduced glutathione, it will be converted to a less harmful, or it will be converted into harmless substances such as water and oxygen. Okay, let me repeat the process. The HMP is a diversion pathway from the Embden-Beyer-Hoff. So the glucose 6-phosphate, okay, when acted upon by G6PD, will be converted into glucose 6-phosphogluconate. Now, the G6PD requires the presence of NADP as its cofactor. So, NADP here is converted into NADPH. And this NADPH is used in the next step. The next step involves the reduction of glutathione. Glutathione is reduced by the enzyme glutathione reductase. In the process of reduction, the glutathione reductase requires the presence of NADPH as its cofactor. So NADPH here becomes or is converted to NADP. And in the process, glutathione becomes reduced glutathione. And these reduced glutathiones are the ones that will counter the peroxides, for example. So in the presence of peroxides or other oxygen-free radicals, reduced glutathione will convert these peroxides into water and oxygen. And these water and oxygen are now harmless to the cell. So that is how the hexose monophosphate pathway prevents the oxidation of hemoglobin. The G6PD provides the only means of generating NADPH for glutathione reduction. So please take note of this. It is only the glucose 6-phosphate dehydrogenase enzyme that is capable of generating NADPH that will be used by the cell for the reduction of glutathione. So in the absence of G6PD, okay, the erythrocytes are particularly vulnerable to oxidative damage. Likewise, normal G6PD activity is able to detoxify oxidative compounds and safeguard hemoglobin, sulfhydryl-containing enzymes, and membrane thiols, allowing normal functioning RBCs to carry enormous quantities of oxygen safely. Remember that the very function of our RBC is to carry oxygen, okay? Now, the presence of oxygen there may cause oxidation of the hemoglobin. But because of the hexose monophosphate pathway, the oxygen is not able to oxidize the hemoglobin. Okay, because of the protective mechanism brought by the hexose monophosphate pathway. Again, the hexose monophosphate pathway is used to generate NADPH, and this NADPH will be used for the glutathione to be converted into its reduced form. And it is the reduced form of the glutathione that will counteract the effects of the oxygen radicals that may potentially cause damage to our hemoglobin. Now, if there will be oxidation of the hemoglobin, particularly the globin chain, okay, it will result into the formation of Heinz bodies. Now, what are these Heinz bodies? Heinz bodies are, or they come from denatured hemoglobin. Okay, when the hemoglobin is denatured, okay, particularly the globin chain, when the globin chain of the hemoglobin is denatured, it will be precipitated in the cell. And once it precipitates in the cell, it will manifest as Heinz bodies. And Heinz bodies look like this. They look like dots on the membrane of the RBC. Now, please do take note of this. Heinz bodies are only visualized using supravital stain. As discussed in your histopath, supravital staining involves staining of the cell while the cell is still alive, okay? Because the cell has to uptake the stain, and once the stain is diffused inside the cell, the stain will be able to stain what the internal structure of the cell. So Heinz bodies are only visualized using supravital stain. You cannot visualize a Heinz body using the typical right stain. Right stain is what we use in the laboratory for staining blood smear. Okay, when we view blood smears, right, that it is the right stain that we use, no, to visualize these cells. But when we use right stain, we cannot visualize Heinz bodies because again, Heinz bodies are only visualized using supravital stain. Example of supravital stains are that are used for our RBCs are methylene blue and uh, cressyl violet. Okay. Now, why are we discussing Heinz bodies and what does this have to do to the function of the RBC? Okay, class, please remember that your RBC contains hemoglobin, and what should be present in the cytoplasm of the RBC are only the hemoglobin plus the enzymes plus the electrolytes in the metabolic products of of the RBC. If there will be other foreign bodies such as these Heinz bodies, Heinz bodies are, by the way, considered as foreign because these Heinz bodies come from a defective hemoglobin. Okay, when these Heinz bodies precipitate on the RBC membrane, um, the RBC membrane with the Heinz bodies become rigid. Okay, let me repeat that. The portion of the RBC membrane with the Heinz bodies become rigid. And when they become rigid, as the cell passes through the spleen in the splenic sinusoid, again, the splenic sinusoid have a very small passage area. But if a normal RBC can just squeeze through it. But if the RBC has a rigid membrane, it cannot pass through the splenic sinusoid. As in the case of Heinz bodies, only a portion becomes rigid. So in this case, okay, the RBC here becomes trapped because of this rigid portion here. The trapping of this portion of the RBC here activates the splenic macrophage. The splenic macrophage will just take this portion. Okay, it will remove this portion only, not the entire RBC because only this portion is damaged. So after taking out this portion of the membrane here, the cell becomes like this. Now, this cell has a portion that is bitten, right? Now, this cell is what we call as bite cell because, well, they really look like they have been bit, right? So these bite cells, okay, they look like this in the when stained with right stain, okay? So this portion here indicates that something has been removed, but only this portion here is removed, not the entire cell, because the other parts of the cell is still normal. The other parts of the membrane is still normal. Only this portion is abnormal because only this portion is rigid because of the presence of the hind bodies. Now, if you may ask, is this cell still capable of transporting oxygen? Yes, this cell can still transport oxygen, however, not as efficient as the normal RBC with an intact cell membrane. Okay, so eventually this cell will be destroyed because of its loss of its viability. Okay, and if there will be abundance of Heinz bodies in the membrane of the cell or in the cytoplasm of the cell, that will eventually cause the destruction of the cell. So again, good thing we have the hexose monophosphate pathway that will prevent the oxidation of the hemoglobin. Okay? Now, if you may be wondering what happens to those individuals with G6PD deficiency. Okay? I'm sure you are already familiar with G6PD deficiency because you have already encountered this in your MT loss, right? Particularly in newborn screening. This is the reason why we include G6PD deficiency in the newborn screening because so that as early as newborn, there will be appropriate management in the case of the child. Okay, the individuals with G6PD deficiency, they don't really have a terminal sickness. Their condition is manageable. All they have to do is just to avoid foods and other exposure, and they have to avoid food and other exposure to that will generate oxygen radicals. So there are certain lists of food that they have to avoid in order to prevent excessive production of oxygen radicals. So they can still live a longer life as long as they don't expose themselves to oxidative stress. Okay? Now, another diversion pathway is the methemoglobin reductase pathway. Okay? Now, okay, before we proceed, allow me to discuss this for a while. The functional form of iron is in the ferrous state. So the functional form of iron which is found in the hemoglobin is in the ferrous state. Okay, please take note of that. The iron in the hemoglobin is in the ferrous state. But when the iron is oxidized, okay, it will become ferric state. No? Is there a possibility that the iron in the hemoglobin becomes oxidized? Yes. There is a very big possibility that the iron inside the hemoglobin will become oxidized, particularly if there are abundance of oxidants in the environment of the RBC. Now, in order to prevent the oxidation of iron, okay, this is why methemoglobin reductase pathway is important because the methemoglobin reductase pathway will prevent the oxidation of iron. So in other words, it will maintain iron in the ferrous state. Now, you may be wondering, Ma'am, what happens if the iron is in the ferric state? Why? No? What does this have to do with the RBC? Once the iron is oxidized into the ferric state, the hemoglobin will not be able to bind nor to unload oxygen. So in other words, when the iron of the hemoglobin is oxidized in the ferric state, the RBC will not be able to perform its function. And if there will be enough poisoning that can cause massive oxidation of the hemoglobin of the iron of the RBC that can cause to cyanosis. Cyanosis occurs when no oxygen is delivered to the tissue. So there will be extreme hypoxia. The patient becomes blue in color because of the lack of oxygen. And again, the depletion of oxygen there is because the RBC is not able to let go of the oxygen. It was not able to let go of the oxygen because its iron becomes oxidized. Okay, so it is important that for RBC to let go oxygen, its iron must be in the ferrous state. Okay, so what happens in the methemoglobin reductase pathway? Now, again, every day we are exposed to oxidation. Now, when oxidation occurs, the iron in the hemoglobin may be oxidized into ferric state. Now, you may be wondering, Ma'am, what happens to we have the HMP pathway? Okay, the glutathione in the HMP must be able to reduce this iron back to the ferrous state. But no, the HMP pathway, the glutathione, the reduced glutathione in the reduced glutathione is not able to convert ferric iron into ferrous iron. Okay, it doesn't have that capacity. That is why it is important that RBC has to undergo this methemoglobin reductase pathway, because this methemoglobin reductase pathway is mediated or is catalyzed by the enzyme methemoglobin reductase. Okay, methemoglobin reductase will perform the reduction process. And in the process of reduction, it donates hydrogen. Okay, and as it donates hydrogen, the ferric iron is converted into the ferrous iron. Now, NADPH may also participate in the process. Okay, the NADPH generated in the HMP pathway. It may participate in the process of reduction here, but NADPH, it takes, um, NADPH reduction takes very slow. Okay, methemoglobin reductase is more efficient when it comes to reducing iron into from the ferric state into the ferrous state. Okay, and ADPH here will assist, may assist, okay, but it won't be able to reduce iron as much as methemoglobin reductase pathway. Okay, methemoglobin reductase is otherwise known as cytochrome B reductase. Okay, now you may be wondering, Ma'am, where does the H come from? How come there is H here carried by the methemoglobin reductase? The H here comes from the NADH generated during the conversion of glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. This is still the conversion of glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate is part of the Emden-Meyerhof pathway. So during this process of conversion, it generates NADH. So NADH here donates the H here to the methemoglobin reductase. And then methemoglobin reductase uses this H here in order to reduce the ferric iron of hemoglobin into the functional ferrous iron, which is in the form of hemoglobin. By the way, when the iron is oxidized, when the iron in the hemoglobin is oxidized into the ferric state, we call the hemoglobin as methemoglobin. Okay? Hemoglobin when the iron is in the ferrous state, methemoglobin when the iron is in the ferric state. Okay? I hope that's clear. Okay, and now we've come to the last diversion pathway. But before we proceed to that last diversion pathway, allow me to describe first to you the typical structure of a hemoglobin molecule. Okay, the hemoglobin molecule. This is the hemoglobin molecule of an adult. Well, basically, the hemoglobin molecule is a tetramer, right? Now, this hemoglobin molecule for an adult, okay, it has two alpha chains and two beta chains for adults. But generally, the template of hemoglobin is it is composed of two alpha chains and two non-alpha chains. And you will discuss in your next lecture in hemoglobin what are those non-alpha chains. Okay, but for the sake of discussion for this topic, allow me to use the predominant hemoglobin in an adult, and the predominant hemoglobin in an adult is the hemoglobin A1, or also known as hemoglobin A. The hemoglobin A, or A1, is composed of two alpha chains and two beta chains. Okay? Now, in each chain, the chain, by the way, is the globin chain, the protein portion of the hemoglobin. Each globin chain contains one heme molecule. Okay, as you can see. So together, this tetramer is called or comprises the one hemoglobin molecule. Now, the very function of the Lou-Behring Rappaport pathway is for the regulation of oxygen release in the tissues. And how is it possible? The Lou-Behring Rappaport pathway is responsible for the generation of the 2,3-DPG. Okay? Before we discuss the mechanism of this 2,3-DPG, please watch this short clip here. Okay? As you can see, the hemoglobin is able to take up oxygen, because basically that is the very function of the hemoglobin, to take up oxygen and to deliver this oxygen into the tissues. When hemoglobin is bound to oxygen, the molecule is called oxyhemoglobin. But it's not forever that oxygen will be bound to hemoglobin, because oxygen has to be unloaded and to be delivered into the tissues where it is needed. So the unloading of oxygen causes the beta chain of the hemoglobin to be pulled apart, as in this presentation here. Now, this pulling apart of the beta chain allows the entry of 2,3-DPG. The entry of the 2,3-DPG, or 2,3-DPG being bound to hemoglobin, allows the release of more oxygen into the tissues. Basically, it's like this. Hemoglobin has high affinity for oxygen. So under normal circumstances, oxygen will diffuse into the cell and will bind with hemoglobin effortlessly. Because of the high affinity of oxygen to hemoglobin, hemoglobin may not release oxygen readily. So that's when 2,3-DPG comes in. Your 2,3-DPG will interrupt the affinity of hemoglobin to oxygen. In other words, 2,3-DPG decreases the affinity of hemoglobin to oxygen. When hemoglobin has low affinity for oxygen, it can readily release oxygen into the tissues. So in other words, when 2,3-DPG is bound to the hemoglobin molecule, oxygen will be readily released as needed by the tissues. Now, when the 2,3-DPG is bound to the hemoglobin, actually the attachment here is because of a salt bridge that forms between the beta and the beta chain, as in this presentation here. So the salt bridge formed between the beta and the beta chain allows the attachment of 2,3-DPG. When 2,3-DPG is bound with hemoglobin, the hemoglobin is in a tensed state. Okay? When 2,3-DPG is attached to the hemoglobin is in the tense state. Now, this tense state of hemoglobin is, of course, deoxygenated, because the oxygen are already unloaded in the tissue. That is why 2,3-DPG comes in here, right? So this deoxygenated hemoglobin will go to the lungs in order to uptake oxygen for another round of delivery. Now, in the lungs, there is increased oxygen tension. The increased oxygen tension in the lungs causes the salt bridge to dissolve. The dissolution of the salt bridge will dissociate 2,3-DPG from being bound with hemoglobin. So in other words, 2,3-DPG will be out of the picture because of the increased oxygen tension in the lungs. And when 2,3-DPG is already out in the picture, okay, the hemoglobin is now free to accept and to bind with oxygen. So in other words, in the lungs, okay, in the lungs, the oxygen can bind once again with the hemoglobin after 2,3-DPG is removed from being bound with hemoglobin. Okay? Now, when oxygen is being bound to hemoglobin, the hemoglobin confers a relaxed state. Okay? Now, this relaxed state of hemoglobin will go to the tissues in order to deliver and unload oxygen. And the same process is repeated. Okay? Now, what happens when there is hypoxia? Hypoxia occurs when there is decreased oxygen tension in the tissues. The decreased oxygen in the tissue, of course, it means that there is increased oxygen need. When there is increased oxygen need, RBCs have to double their effort in terms of oxygen delivery. So, as a compensation, your RBCs should be able to deliver more and more oxygen at a faster rate than than the normal state. Now, when hypoxia occurs, there will be decreased oxygen hemoglobin affinity. This decrease in the oxygen hemoglobin affinity will allow the release of more oxygen in the tissues. Likewise, hypoxia also drives glycolysis to increase. When there is increased rate of glycolysis, there will be more ATP to produce, more 2,3-DPG to be generated, and an acidic pH. The drop in the pH causes a decrease in the oxygen hemoglobin affinity. So in other words, an acid pH will decrease the affinity of oxygen and hemoglobin and therefore increasing the release of oxygen in the tissue. So there will be two mechanisms that influence the faster release of oxygen in the tissue. First is to increase the concentration level of 2,3-DPG because the more 2,3-DPG there are, the more oxygen will be released into the tissues. The second mechanism is by decreasing the pH. When glycolysis is at a faster rate, the pH of the RBC will drop a little. So that drop in the pH, an acid environment is created. An acid environment decreases the affinity of oxygen to hemoglobin. Therefore, oxygen will be readily released into the tissues as needed. Now, you may be wondering if 2,3-DPG becomes depleted. Well, not literally depleted, but it becomes saturated. Free 2,3-DPG becomes saturated and becomes bound to hemoglobin. So like more of a depletion of the free 2,3-DPG. Now, this state of fully saturated 2,3-DPG will increase more the rate of glycolysis in order to generate more 2,3-DPG. So, the generation of more 2,3-DPG is done in order to compensate to the increased need, no? And remember that if there will be more 2,3-DPG, okay, there will be more oxygen release. There will be more oxygen that will be released into the tissues. But you have to remember also that there will come a point that the need exceeds the capacity to compensate. Yes, your RBCs will be able to compensate. Yes, your bone marrow will be able to produce more RBCs. But if the cause of the hypoxia is not addressed, that need will exceed the capacity of your body to compensate. And when such happens, the patient's life will be put into jeopardy and may lead to the death of the person. So, it is important that doctors should be able to identify the root cause of the hypoxia because at some point the RBCs will not be able to compensate and to respond to this need, especially if the need is at an extreme level. Okay? Now, this is a summary of the different metabolic pathways that RBCs perform in order to carry out its function properly and to serve the entire lifespan of the RBC. Again, if there is an abnormality in any of these pathways, it will result into the loss of the viability of the cell. It will result to the loss of the deformability and flexibility of the cell, and it will result into an early destruction of the cell. Now, next question will be what happens to the RBCs when they reach senescence? Senescence is old age. Remember that RBCs will serve a lifespan of about 90 to 120 days. So when RBCs reach that 90 to 100 RBC, 90 to 120 days, of course, they become aged. So what happens to an aged RBC? Will there be a change in the characteristics of the RBC? Will there be a change in its shape, in its deformability, and whatnot? Yes, of course. When RBCs age, there will be changes in the property of its membrane. So therefore, there will also be changes in its efficiency in terms of delivery of oxygen. Now, an aged RBC, its membrane becomes less flexible, and when its membrane becomes less flexible, it will be trapped in the splenic sinusoids, and trapping of the RBCs in the splenic sinusoid will activate the splenic macrophages, and the splenic macrophages will destroy those trapped RBCs there, and therefore causes hemolysis or destruction of the RBC. Another that will occur is that the concentration of cellular hemoglobin increases. Now, is this a good thing? Not really. When hemoglobin increases inside the cell, that causes a change in the shape of the cell. Remember that a cell, the RBC, has to maintain its biconcave shape, and most of its hemoglobin is concentrated on its periphery in order for an easy and faster rate of oxygen uptake and unloading. But if there will be increased hemoglobin concentration inside the cell, that will change the shape of the cell. From being biconcave, the cell will become spherical. And remember what happens when the cell is spherical? Yes, it loses its flexibility and deformability, and that kind of cell is subjected again for hemolysis. Okay. And lastly, enzyme activity diminishes when the RBCs age. Of course, when enzymes and their activities will diminish, the different pathways, energy pathways, the HMP, the methemoglobin reductase, and the Lou-Behring-Rappaport pathways will also be affected. So there will be a decrease in the metabolic activity of the cell, and a decrease in the metabolic rate of the cell will cause a change or a loss of its flexibility and deformability, a change in the shape. In other words, when the enzyme activity of the RBC diminishes, the RBC will lose its viability also. And when RBCs lose its viability, they are subject for hemolysis or destruction. But again, this whole thing is a normal process because the RBCs have already reached their lifespan. They have already served the 90 to 120 days lifespan. So it's time for these RBCs to rest. They have done their job well, and it's time for them to be destroyed. But when they are destroyed, though, they are not totally destroyed because some of their parts are being recycled. Okay, as it was discussed to you in the destruction of RBC, remember the contents of the RBC will be released, releasing the hemoglobin molecule, and then this hemoglobin molecule will be broken down further. The globin will be recycled and will be brought to the globin pool because this globin is protein, and proteins must be recycled. The heme, however, will be degraded into bilirubin and finally to urobilinogen. Okay, and that will be the end product of heme degradation. Okay, so basically, this is what happens. Please do take note that there are about 10% of the RBCs that are hemolyzed prior to their old age. That 10% hemolysis accounts for RBCs that are damaged. Okay? Now, for in a normal scenario, an RBC has to go through the 90 to 120 days lifespan, and when they become aged RBCs, when they are already senescent, that's when they are being destroyed in the spleen. So in the spleen, it's the macrophage that cause the destruction of these RBCs. And then RBC will release its content. The heme in particular will be broken down into bilirubin, And then this bilirubin will be broken down as if into urobilinogen. Okay, and urobilinogen will be excreted via the urine or via the stool. When urobilinogen is excreted via the stool, it will be in the form of stercobilinogen that when oxidized becomes stercobilin. And stercobilin is responsible for the brown coloration of your stool. Whereas for the globin chains, these are basically proteins, so made up of amino acids. These amino acids will be brought to the globin pool, okay, in the bone marrow because these amino acids will be once again recycled in generating another batch of RBCs. Iron also, iron is not it is not eliminated out of the body because as much as possible iron has to be recycled, and that's the normal mechanism of your body to recycle iron. So iron will be brought to the iron pool in the bone marrow, and that iron will be used in creating or in generating another batch of RBC. Okay, so that ends our discussion. I hope you are able to get what I'm trying to or rather, I hope I am able to convey to you the necessary information that you need in order to understand the RBC kinetics. Why is it important for RBC to have a flexible and deformable cell membrane? And what contributes to this flexibility and deformability? What factors influence the shape of the RBC, and why is the shape of the RBC important for its function? And the different energy metabolic pathways that RBC have to perform in order to sustain its viability. So thank you so much for your time and for listening. I hope that you'll be able to remember all of this information. So see you in our next lecture.